My name is Dale Overton. I'm the CEO of Overton Environmental Enterprises. We're a company that manufactures advanced ecologically based products for agriculture. And then we also have an industrial division that manages organic wastes for large food processing companies. Today, I'd like to talk about soil biology, the science behind it, and its implications in modern agriculture. I think that there's been a paradigm in modern agriculture, and that is soil has been depleted of its organic matter over the last 50 years with the advent of modern conventional ag. It's basically just been considered dirt and been completely mismanaged. Microbes that have formed relationships with plants and soil over the past thousands of years have been ignored, and their roles have been discounted for modern day fertilizers. And what this has done is created basically a massive agri-industrial complex that completely discounts what Mother Nature has naturally done to help plants and agri-systems in their natural form grow. Typically, when you look at the modern agricultural fertility programs, pH is the real driver. You look at the nutrient pool in the soil, you determine what the pH is, and that gives you your availability of nutrients. It's all chemistry. It tells nothing about what the plant is doing or what the actual life in the soil is doing for the plant. The other thing that people look at is something called Mulder's chart, which is how the availability and presence of certain minerals will impact other minerals. So for example, you've got excess potassium in your soil, you'll see an imbalance in magnesium and calcium. However, this again doesn't tell you anything about what biology is doing. It strictly talks about what's happening chemically in the soil. Soil is not just dirt, soil is alive. It has an immense functional role in the global ecosystem. Again, if you consider a plant, it's got 50% of its biomass is actually under the soil in that root mass. And that's responsible for almost all nutrient uptake. You get some from the foliage, but mainly your nutrients are taken up from your root zone. Soil houses a huge functional diversity of microbial life. We know more about the moon than we know about soil. So I think it's really important that we spend time looking at what soil is doing and what kind of organisms live in the soil and how we can harness that information and use it to our advantage to help reduce the amount of fertility we're adding into our crops, maintaining yields, but also just creating a sustainable food system. How are those plants getting their nutrients? How is that biomass created? Well, it's created through the activity of microbes and through microbial movement, growth, and development. Nutrients are released. There's a whole plethora of roles that microbes basically have in an ecological system. Here, what we're looking at is a schematic of a soil colloid with some root hairs, bacterial colonies, mycorrhizae. You've got your actinomycete bacteria, you've got nematodes, flagellates, you've got your clay and organic matter complex, and you've got basically this soil aggregate that's got good airflow and good microbial life. This is what you want to see. Ultimately, most of this action is happening in the root zone. So as these microbes are doing their thing, they're releasing nutrients through mineralization of organic matter, but also through their activity in the form of feces. So if you really take a look at what healthy soil looks like, healthy soil really looks like cottage cheese. It shouldn't be compacted and full of different crop residues and have very little airflow. If you have very poor airflow, you're going to have very low diversity of microbes in your soil, and your soil is going to lose its ability to function properly. This will never break down. It's got no airflow, there's no fungi. Bacteria might break some of it down, but in doing so, they're going to suck up nitrogen out of the system. In a natural system where you have a lot of microbial activity and things run quite naturally, that oxygen and that airflow really help to create stable soil aggregates. Here's a series of photos here where we're getting closer and closer into a healthy soil aggregate. And what you'll see here is that there are a lot of organic matter, clay, mineral complexes with a lot of airspace. So you can kind of see that there's really good potential for this soil to house a really positive microbial community that's going to be able to access the resources that the soil has to offer. This is our Organics Resource Management Facility in Portage la Prairie, Manitoba. And what we see here is the process of basically building healthy soil. And we do this by taking food processing waste and blending it with various carbon sources to create what we call a humified compost. And this is kind of like the cottage cheese that I was talking about. As you see, there's larger aggregates, smaller aggregates, and there's really quite a bit of life in here. As you can see, there's quite a bit of fungi growing on this product right now as it's in its final stages of growth. So any of the carbon that's left is actually growing fungi on the tips, and you know, the wood chips basically peel apart very easily. So you've got good degradation going on there. Really what I'd like to talk about for the rest of this presentation is how different microbes function in plant growing systems. There's about 5 or 6 different functions that I want to look at, including nutrient mineralization, phytohormone production, antibiotic production, nutrient cycling, and crop residue management. These are the real big things that microbes do that I think are misunderstood, and hopefully we can enlighten the audience on some of these things over the next few minutes. So here we are in a cornfield in southwestern Manitoba, just west of Winnipeg. And this cornfield is special because it's been treated with a biological product. And we've had a very interesting season so far, and this corn actually looks fantastic. It's probably about 10.5 feet tall. You got some really nice cobs on it for Manitoba. What I'd like to talk about next is how microbes have been ignored in modern agriculture. And mainly that's because fertilizers have kind of replaced the traditional role of what microbes have been doing in a soil. You know, they're easier to manage, they're easier to apply, you can see results immediately. So, you know, there's a true rationale as to why people would turn to, you know, synthetic fertilizers for crops. But again, I think that discounts significantly what microbes are doing. So fertilizer overuse, tillage have a negative impact on important functional microbes, and mainly bacteria and fungi. Soil oxidation and mineral export, it's just not sustainable. You know, when you burn your crop residues or you're exporting all your straw and you're not putting any of that back into your system, you're going to be at a net loss. So over time, we're losing organic matter, we're losing mineral concentration in our soil, we're mining them basically, and ultimately you're putting down more and more for less and less return all the time, and it's really just not a sustainable system. Understanding how microbes function in bulk soil is vital to creating a sustainable agricultural solution, or system, I should say. Fungi are very sensitive organisms. Tillage really impacts fungi because they grow they call them hyphae, that grow into a mycelium. And if you till it, you break that up, you're actually essentially breaking or fragmenting those fungi. And when you do it enough, it basically kills them and you lose that function in your soil. 2% of fungi fall into that pathogen category. 98% of all fungi are actually beneficial and have an important role in the ecosystem. One of the reasons microbes have really been ignored is because we can't see them. There's new techniques that have really kind of unlocked the true secrets of the microbial world, and these techniques are called metagenome analysis or genetic analysis. So we can actually understand the who, the whats, the whens, the wheres of the microbial world, and how then can we use that information to influence how we farm our crops and our fields. We can now take a sample of soil and we can look at the DNA of all of the different bacteria and fungi that grow in these soils and get an outlook of what that soil looks like from a diversity standpoint. You can code for phosphorus solubilization or phosphatase, which is an enzyme that pulls phosphorus out of solution. to help understand and benchmark soil. So you can actually say, we know that this soil has this ability to remove phosphorus from the locked up organic matter because it has a lot of this phosphatase enzyme, right? The gene that codes for it is present. And again, this helps to isolate and discover functional microbes that aid in plant growth, disease resistance, and development. It's basically bioprospecting. And it's what the medical field is doing to look for new antibiotics. It's what large agricultural companies are doing to try to isolate certain microbes that can, ultimately be patented or form some kind of intellectual property, and then they can sell to you, obviously. I'm more of an ecological fan myself. I believe that it's more about a system than one microbe. So that's why we do what we do, but happy to chat about that with anybody. So this photograph is really of the bacterial community that we've isolated from our product called EcoT. So this is a metagenome analysis done by Mr. DNA out of Texas, and this was part of a master's program. And what we can see here is we have a whole bunch of functional bacteria along the bottom axis, and then we have different products that we build at our company, and it basically shows which products these microbes basically come from and how we can utilize techniques to grow and encourage their growth and development using a bioreactor. So it's very, very cool, and this is really what this whole future of metagenome work is allowing us to do. We can understand, using genetic techniques, exactly how our soil functions microbially, so we can augment the microbial community with fertilizer and allow the soil to feed the plant, and not necessarily the other way around. Once we can do that, I think that ultimately we will create a much more profitable farming system for the modern farmer. If we take a look, the corn is, it's not sending out any tillers. These are really nice long tap roots. You've got these fine roots. So these roots here are your water roots, and then these finer roots are really the homes of the microbes. And ultimately, that soil root microbe interaction is kind of where everything's happening. And when you have a really healthy community, you'll see that this just falls apart really nicely like cottage cheese, and that's a really nice soil structure. Ultimately, this is a really hard clay soil, but right in the rhizosphere, right where the plant roots are, you see it's really like cottage cheese, and that's what we want to see. And that's the signs of a really healthy soil. And ultimately this is what we want to do, because when we have this, we can really start to reduce the amount of inputs that we're applying to this crop. Now in this case, I'm not sure if this had a fungicide or not. I'm sure that there was a pre-emergent herbicide on it, and again, I'm not preaching don't use fungicides or herbicides. What I'm trying to say is if we use biology, we can use those products less and less, and I think that's really the take-home message, is ultimately to create that sustainable system. We need to rethink about how we use these tools like the fungicides and insecticides and synthetic nutrients. For example, anhydrous ammonia is a well-known bacterial killer, as is glyphosate. So, again, I think that by understanding the roles microbes play and the value they truly have in the system, we can all do better. We have a system, we know that it's crashed, we want to bring it back into a healthy point. We call this, in a way, we have to now be ecological engineers. We have to now engineer our soils back into a position where we can get really healthy crops. We can understand how soil types and planting regime influence biology. Rhizobium is a great example. You don't want to plant peas into a field that you've been monocropping corn out of for the past 20 years, because your soil won't have the organism called rhizobium, which is very important for a legume to mineralize nitrogen from the atmosphere. We want to understand what demands each crop has and how microbes can supplement fertilizer, so it's a more holistic approach. When I talk about that, I'm talking about functional diversity. If we're looking at ecological engineering, the definition of ecology is the interaction of all organisms and environment with one another and their environment. So you can't just look at one species, you have to kind of look at it from a systems approach.
And ultimately, when you're using single species inoculants like rhizobium, yes, they're great for soybeans, and yes, they're great for fixing nitrogen from the atmosphere, but they're not necessarily really good at mobilizing phosphorus or pulling zinc out of a locked up soil colloid or anything like that. So again, we want to get that functional diversity, and microbes really can bridge that gap, especially as it pertains to micronutrients.
We want to use microbes to improve plant growth and development. So we're trying to engineer that system or that rhizosphere community towards what that plant needs. And that plant will also select for those microbes as well by releasing exudates and sending out signals and whatnot. The other thing, we want to use microbes to recycle crop residues or trash back into the soil. So again, instead of exporting it or burning it off, we can use microbes to actually reintegrate that back into the nutrient pool and organic matter in the soil.
Music bed.
Microbial roles in plant growth and development. Basically, plants provide sugars through exudates to the soil, which stimulate